Assalamualaikum and hello everybody. Today our group are going to present about the analysis of zebrafish contamination with heavy metals using full field X-ray fluorescent imaging system based on a MEGD. Okay, now let me introduce myself. My name is Muhammad Aziz Hakimi bin Zaidi, and the person that will be presenting with me is my friend Muhammad Ramzi bin Kamaruzaman. Now, why is it important for us to learn about this research? Well, heavy metals can harm aquatic life and it can also enter the human food chain, which is not ideal. So, understanding how and where these metals accumulate in the fish help in risk assessment for the ecosystem and public health. These zebrafish are often used as bioindicators. Detecting heavy metals in their tissue helps assess pollution levels in aquatic environments.
Now we are going to talk about alternative of full-field X-ray fluorescence spectroscopy, which is micro X-ray fluorescence with silicon reef detectors. Why we are thinking using this as alternative? Because it offers high energy resolution, which is 125 to 150 electron volt, allowing better separation of overlapping peaks like zinc and copper. Next is for its trace element sensitivity because it can detect very low concentration of metal, ppm or ppb range. Next, it is better elemental discrimination. It reduces spectral overlaps, improving quantitative accuracy. Next, it has focus beam, which means it has high spatial resolution. It can achieve below 20 micrometer spot size using polycapillary or pinhole optics. And lastly, it is well established for biological tissues and it is frequently used in toxicology and bioimaging. Now we are going to discuss why micro X-ray fluorescence was not chosen and instead it is full-field X-ray fluorescence that was chosen for the case study because micro X-ray fluorescence has slow point-by-point scanning. Micro X-ray fluorescence requires faster scanning which takes much longer over wide area. Next, it has high cost and complexity. SDD-based systems with X-ray optics are more expensive and complicated to operate. The third one is beam damage risk. Focusing X-rays can increase the local dose and potentially damage sensitive biological tissues. The fourth one is limited field of views because only small areas can be analyzed at high resolution, not ideal for full body mapping like zebrafish. And lastly, because of sample manipulation, precise positioning and flat samples are often needed for the micro X-ray fluorescence, which making it adding prep complexity.
So now we move on to full field XRF spectroscopy. So this spectroscopy has two results, which is from the spectra and also the distributed mapping. So the spectra shows the energy peaks that correspond to different elements. Each heavy metal has a unique characteristic energy lines, whereas for distributed mapping, it is a way to show an image based on a specific energy range that has been selected. It also produces 2D image showing the element's localization. So now we see two images. On the left, we can see that the spectrum produced by XRF have three significant peaks, which indicates the three metals that are present, which are calcium, zinc, and manganese. The higher the peak shows the higher the concentration of that metal. Now we move on to the right image, which shows three different image distribution mapping for each metal. We can see that the metals location throughout the fish and how spread apart are they from each other. We can see that the calcium are the most spread out due to the fact that the fish bones are made out of calcium. Each peak represents a heavy metal observed by XRF. By matching the energy range, the element's identity can be determined. The height of each peak indicates the concentration of the element present. Although the spectrum gives a lot of information, at certain times it may be insufficient. Therefore, distributed mapping, by matching the energy range, must be used in order to conclude its presence. Although we can see that selenium exists in the spectrum, it only appears as a small peak. Therefore, we need to determine whether that small peak is the presence of SE or is it simply just background. So we must verify it by using elemental distribution map. How does this work is by revealing a clear accumulation, which the color green in the specific region, confirming its presence although low concentration. This supports the conclusion that selenium is present at a potentially concerning level. So now we move on to the quantitative analysis. So how is it able to detect the concentration of the heavy metals? Well, the zebra fish embryos are exposed to the known concentration of heavy metals from zero point 0.1 to 5 milligram per liter. Then the biological response are recorded, such as mortality, heart rate, body length, and deformities of the embryo. Then those response curves are plotted and used to determine the toxicity level. After that, the LC50 or lethal concentration for 50% of the embryos is calculated. As you can see, the result shows of the three metals which at that concentration can be considered as highly toxic. Now we move on to the limit of detection, or for simple term LOD. This formula helps us determine the smallest amount of substance of heavy metal that can be reliably detected. So there are three things in this formula, which are concentration, background signal, and the next signal of the sample. So in the table, we can see the metals that have been shown their limit of detection value, and which means that we can just detect them quite easily, and it can ensure that it is sensitive to even a small traces of the element.
Next is the advantages of full-field X-ray fluorescence spectroscopy. The first one, it is simultaneous multi-element detection, which means it can identify several elements at once without destructive sample treatment. Secondly, it is non-destructive and minimal sample prep. There is no need to alter or destroy the specimen, which gives simple preparation and preservation of zebrafish. The third one is it is fast wide area imaging. It captures an entire field at once instead of point by point scanning, and will greatly reduce analysis time. And it is lower complexity and cost. It is simpler and cheaper to build compared to micro scanning system using silicon detectors. It is also high rate and operating at room temperature. The gases 2D detectors works at ambient condition and handles high count rate. It is also good spatial resolution. It can achieve around 50 micrometer to 400 micrometer across larger areas depending on the setup. And lastly, it is suitable for biomedical mapping, which is it is well suited for identifying element distribution, for example, mangan, selenium, or lead in tissues like zebrafish. Next part is the limitation of full-field X-ray fluorescence spectroscopy. The first one, it is moderate energy resolution. Gas detectors offers far lower energy resolution, which is below 1 kilo electron volt, compared with silicon CCDs, which is 150 electron volt, complicating separation of nearby element peak. The second one is it is not optimized for trace detection. Gas detectors have lower detection efficiency at higher energies and may miss very low concentration elements compared to more sensitive system. The third one, it is limited in depth or 3D resolution unregulated. While good for 2D tissue mapping, this setup doesn't inherently provide depth profiling like confocal or tomographic techniques. The fourth one is the spectral overlaps and identification challenges. Lower resolution can lead to misassignment, for example, copper versus zinc, especially with complex spectra. And lastly, complex data analysis needed. Uh, it is often requires advanced methods like non-negative metric factorization to disentangle overlapping spectral signals.
And now we move on to suggestion and improvement that can be made. One of them is by using heavier noble gas such as xenon and krypton, which greatly enhances the system's sensitivity, especially for elements like Xe, whose current detection efficiency is quite low. Other than that, the use of multi-hole collimators could reduce acquisition time, improving image quality, and minimize background noise or pixelation, making the system faster and more efficient for broader application.
For the conclusion, full-field X-ray fluorescence spectroscopy successfully visualized metal distribution such as mangan, selenium, and lead in preserved zebrafish with minimal preparation. Next, the method is fast, non-destructive, and suitable for full-body elemental mapping. Though it has moderate energy resolution, it remains effective for general elemental screening. And lastly, result has demonstrated that full-field X-ray fluorescence spectroscopy's potential for biomedical and environmental application, especially for monitoring metal bioaccumulation.
Thank you everybody for watching, and if you like it, considering subscribe, and I see you in the next video. Bye bye.